Welcome to the acquisition and analysis session. After excellent talk by Badri sir, we have learned so much about the CD34, that is the stem cell enumeration, and processing video of our colleague. So we will do the acquisition now, and in acquisition we are going to tell about the how to acquire the stem cell enumeration sample, as well as how to do the control, and post that we will do the analysis on Colusa. So in our lab we are using stem kit from the Beckman filter, as well as stem cell enumeration kit from the BD. Before going to for acquisition, so we will revise the thing what we have learned till now. In Badri sir's presentation, so we have learned that the CD40, 45, 34 antibody we have to add to the sample, then incubation, then lysing, and then acquisition. So in the similar fashion, so we are going to do the stem cell control, where we add the normal blood cells 100 microliter with the reverse pipetting. Then we'll add the stem cell control 20 microliter to the tube. We do not add 7AD at all for the samples like controls, which are preserved samples, even if for the UK aquasa as well as the CAP sample.
Sir, I have a question. Why are we not adding 7AD in stem troll?
Well, it's a good question. So, if you add a 7AD to the preserved sample like stem cell control as well as UK aquas and the CAP sample, so you are going to lose the population of interest where your CD34 events comes in the picture if you remove on the basis of viability. So, all the events which are CD34 positive or population of interest are going to a dead cells, and it will give the 7AD positive. So adding the 7AD is as good as you are not adding anything. So to remove the error, like minimize the error, do not add the 7AD to the sample.
I have one more question. How frequently do we run this stem troll control?
So actually, as per the ICH guideline, whenever you perform the CD34 like stem cell enumeration test, you have to perform stem cell control, which is a known value of your sample control. We should be run at the time of before the test. So before acquisition, while our sample is being processed, so in between we have to make sure that our instrument is getting ready. So for this instrument, we have to get the daily maintenance, which has a clean with the vaccine as well as catalyst and then water. After that, we have to perform the daily check, that is calibration, with the CS and T builds. So before acquisition, we have to make sure that our QC is passed. So we'll go to the CST and module for the performance of QC report. So this is the report of our daily QC like calibration, in which you can see these parameters for PMTs as well as target channel, as well as you can see the pass or fail comment. So this will fail when the delta PMT is more than 50. Even it will check for the robust CV for different bits which is less than 6. So after this, I have confidence to acquire my sample as well as the control. So now I will move to the acquisition software. So I have to close this first. So this is our acquisition software, which is a FAXDIO software. One thing is very important: before acquisition, you have to Hotex your tube on the Hotex mixer. So in this, we will make an experiment first. So I am going to take an experiment, and routine standard setting is required for this assay. It does not require any additional setup. If you want to, you can have using the single antibody as well as beads for their location. So we go to the assay, and we have already set up. So I am going to set up which already set up as a CD34 count. So after that, we have template already made for all the plots required for stem cell enumeration. In this, you can see here. So predefined template is ready. Label the assay, either it can be a stem troll or the patient sample which has received from the BMT. So samples are like two types, it can be a pre-PBSC or a harvest. So pre-PBSC we are using direct sample if the count is less than 40,000 per microliter, and for harvest we have to dilute accordingly to match for the your kit. So we have labeled this specimen as well as our experiment. So before starting the acquisition, we have to make sure the parameters required, then the threshold value, we have kept it on the FETC. After that, I will come to the acquisition window. You can see here the acquisition window where we have to define the stopping gate. So my stopping gate is as a singlets bits. So now we will load the sample, that is temporal control for the acquisition. So this is the loading on the SIP, and then we'll acquire. I will start my acquisition, but I am not going to record anything. So in this you can see here, so you should know where your populations are fall. So you can see this plot. So this is my guidance plot where I can see the fault scatter and surge scatter within, all the populations are within the range, within the linearity basically. And we have one more plot which is 45 versus side scatter, which shows me this is also guidance plot. So once I make sure everything is on the place, so I am going to record it now. So I will say record it.
So what are we using for our threshold? Is it CD45 FITC or forward scatter?
No, threshold you should not put on the forward scatter, because you can you lose your bits because they are smaller in size. So you can see here on the plot. So our beads are like here. So if you put it on the forward scatter, you can lose the beads. So always you should keep it on the CD45. That is, we have kept it on the FITC because our antibody is on the is in tagged with the FITC. That's because we don't want any RBCs to be interfere in our acquisition as well as in the analysis. If you acquire all these events, even except putting the threshold, you are end up with a huge number of events as well as a large data file, so that you cannot also acquire the thing.
So, how many events do we need to acquire?
Yeah. So as per the ICH guideline, total number of events should be 75,000, as well as your HPC, that is hematopoietic stem cells gated, true gated should be 100. But in our setup, what we do, we have set up the acquisition for the singlets bits, that is 5,000 number, as well as your CD34 events should be more than 200. So this is because to increase the sensitivity of the assay. So we are acquiring our stem troll control. So this is almost is being done. So meanwhile it is getting recorded. We'll discuss this plot as well as basically ICH protocol with the Boolean gating here. So you can see here the plot we have added, which is FSC versus time, to know whether how is the flow rate and our acquisition rate and our acquisition. It will tell the irregularities or acquisition issue. Next is the singlets, where we want to remove the doublet. As you can see, there is no doublets events, but this is also a guidance plot where it will show you the populations starting. Like you can see here, the lymphocytes are here. Okay, so these are the lymphocytes, then beads. Next clause is viability, as we do not add the viability dye in this, as all the CD34 events will be dead cells. So this is our one criteria where we fulfill the viability. Next to that, forward scatter, side scatter. This is again the guidance plot. So after viability, we have taken the viable cells in this, which is the CD45 versus side scatter. Here we have gated CD45 events, like all leukocytes, true leukocytes here. Third plot is CD34 events, which is side 34 versus side scatter, and gated on CD45 positive WBCs. After that, a fourth plot, which is CD45 and side scatter, to fulfill the criteria low side scatter as well as 45 dim, which is not acceptable. We are done with the acquisition. Very importantly, you should know, in BD software, you should take care of the hierarchies, which gate is coming from which gates. So this is a Boolean gating. You can see here the singlets, viable from the singlets, then lymphocytes, and 45 positive WBC. Out of 45 positive, we have CD34. Out of CD34, we have low side scatter and 45 dim events. Out of that, we have defined our CD34 event based on the lymphoblastogeneric region. So to show our CD34 events falling in lymphoblastogeneric region, we have the lymphocytes, and we have removed apoptic body platelet debris, so that our CD34 events are artifact free. This plot is for the MNC gate, and we have gated our beads on the 45 versus APC. You can get your beads on 45 versus P also, but you have interference of CD34 positive events. That's why this is the best plot to get your beads. And upon this, we have one more plot is added, that is a singlet beads. So temporal acquisition has been done now, so we will unload the tube. So after the acquisition of control, temporal control, and we analyze for it, so our values are within the range, that is 1250 and plus minus percentage of 15%. So now we'll go for the acquisition of our actually sample, that is test sample. So for that we need to make experiment. Again the same thing we'll do. Go to the experiment, then we'll go to the link setup and predefined setup of CD34. Again we'll go to the analysis, template, acquisition template, basically with no specimen. This is done. So labeling of sample should be there. Again we'll go to the parameters, we'll check it. Then we'll check the threshold value as it's previously said while acquiring stem cell control. and then define stopping gate of the singlet beads. So I am going to load the tube now. So for the sample also we'll check for the flow rate. Then this is our guidance plot. So it will say what are the unwanted events and where to cut off value. So limit of acceptance basically. So beads are on the position, everything is okay. So I'll record the tube. So our acquisition has been, I think, almost to the end, because we have captured required amount of beads as well as the total events and our CD34 events. So now the acquisition has stopped, and now we'll remove the unload the tubes. We have acquired our sample for CD34 enumeration in a duplicate as discussed by our colleague in the processing video. So you can do your analysis in the same software, but due to our workload we do not do the analysis in this. So it might, because of the compensation and thing, you might miss the things or you misinterpret the results. So I will transfer this data and this file will analyze in the acquisition analysis system.